My name is Martorana, and my project is looking at the correlation between the copper diet and the growth of amyloid beta plaques and mobility in C. elegans. So my research question: what is the effect of a copper diet on transgenic Canor habitus elegans, also known as C. elegans, growth of amyloid beta plaques, and mobility? First hypothesis, I predicted that the C. elegans that are fed food with the copper diet will have increased mobility and little to no amyloid beta plaques. For my second hypothesis, for the C. elegans that are fed their normal diet with no copper will have reduced mobility and larger amyloid beta plaques. To introduce the background for my topic, I will start with defining Alzheimer's disease. Alzheimer's disease, also known as AD, is a type of dementia and neurodegenerative disease with symptoms like loss of mobility and memory and with an overall loss of independence. This causes the patients to need a caretaker. Some symptoms for AD are tau neurofibrillary tangles, which are the misfolding and tangles of the tau protein, and amyloid beta plaques, which are extracellular plaques found in the brain that are formed from an overproduction of the amyloid beta precursor protein, also known as APP. Often in AD, the copper homeostasis is disrupted in the brains of patients. Copper is required as an enzyme cofactor in many organisms, like in the activities of the mitochondria. This is important to the brain because copper plays a role in synaptic transmission and other cellular processes. The disrupted copper homeostasis can cause neurological dysfunction in the central nervous system. Next, C. elegans are nematodes that are often used as a model organism in labs. This is because they are not resource or time consuming and are easy to work with for vivo experimentation. C. elegans are often compared to humans because they have a genome that 60 to 80% of humans have an ortholog in. Because C. elegans don't express the amyloid beta protein normally, I am using GMC101 strain for my project, which is a genetically modified strain of C. elegans. The importance of my project starts with the fact that copper has been found in higher than normal concentrations in AD patients. If the results of my project show the hypothesis to be correct, the copper diet can be implemented and applied to human patients. This will provide for a more straightforward option for treating AD. My project starts with the preparation of the agar plates. The C. elegans live on agar and NGM, which is nematode growth medium plates. The agar is heated in about 10-second intervals in the microwave until it becomes a liquid and is poured into 6-centimeter petri dishes about halfway, left to cool, and then to solidify. The C. elegans are then transferred to their plates based on their group. The experimental group is the GMC101 strain of elegans that are fed copper. This is done by placing the C. elegans on an agar plate, which is a mixture of a copper solution and the agar solution. The control is the regular C. elegans and the GMC101 elegans, which are fed the regular food, meaning they will be placed on a plate which just has the regular agar. Separation of the elegans is done by chunking. This is when a square is cut out of the original chunk of C. elegans in which they came from and put the square on a prepped agar plate. It takes 1 or 2 days for the elegans to move off of the plate before testing can begin. All the groups of the C. elegans were fed E. coli through a pipette. The plates were then measured underneath a fluorescent microscope to establish the relationship between the copper diet and the C. elegans mobility. Under these fluorescent microscopes, the plaques were measured and the bend assays were performed. A bend assay will determine the copper's effect on mobility. This is where the C. elegans are counted for the number of bends in their body in a minute. The more bends that were seen means an average increase in mobility. I performed 10 trials each day on the control and experimental plates to gather data to analyze. The materials I used for my project were E. coli, C. elegans, both the wild-type and GMC101 strain, NGM, which is the nematode growth medium, 6-centimeter petri dish plates, which is where the C. elegans lived, copper 2 sulfate powder, which was the copper solution used to mix in with the agar mixture for the experimental group, hematoxylin, which is the dye used to view the plaques, and the agar mixture, which is where the C. elegans generally lived. As for my results, the research is still ongoing to develop more accurate results for my project. Research so far shows that there is an average increase in mobility of the C. elegans that were exposed to the copper diet. Some limitations for my project was that it took time to get the wild-type and GMC101 strain of C. elegans from the company that my school was getting them from. Another limitation was the molding of the agar plates, which was caused by the contamination of outside conditions. Both these limitations caused time to come off of my project and less time to analyze data results. Here are some of the references that I used to obtain a background for my project.